Hello, grade 12s. So this is really the last section on DNA, the section of DNA itself, before we start moving on to the sections such as meiosis, genetics, etc. And this is DNA fingerprinting or DNA profiling. So this is really the final bit, the application of DNA technology that we're going to be learning about. Then we'll move on to the other stuff. So before I actually get into talking about what do I mean by a DNA fingerprint and a DNA profile, I need to just cover the difference between what is coding and what is non-coding DNA. So if you think about it, we as humans, we all share pretty much similar DNA. And we know that between different other animals and different other organisms that exist, the further away we get from an evolutionary perspective, the less in common we are going to have. But we know that if we are humans, we are going to be coding for pretty much the same proteins, the proteins that make us who are going to be human. Yes, there will be slight phenotypic differences like the color of your hair, color of your skin, your eyes, a little bit of height, but we are all still human. We look like a human. So that all comes from being able to code for proteins. So now when they were discovering the structure of DNA and they were trying to figure out what was happening, we really only had this focus on the creation of proteins. And back in those days, remember they had limited technology, they didn't really have an ability to understand. They really thought that a small portion of the DNA is actually used to code for proteins. It actually turns out that 2% of our DNA codes for proteins. So back in the day, and even into the late 80s and that kind of stuff, it was figured that a lot of this DNA that was not used for coding for proteins was actually not doing anything in our body. So they just referred to it as junk DNA. So the junk meaning evolutionary junk, as in it is the residuals of the evolutionary process that have kind of stayed in our body but are not actually doing any function for us. Kind of like a vestigial organ, like your teeth got smaller, your appendix got smaller, your whatever it was. We're not using it, but it's still there as part of the evolutionary process kind of thing. So that was why they called it junk DNA. But we now know better. So now what we do, instead of referring to it as junk DNA, we know that it actually does stuff. The other 98% of our DNA does have a function. So we refer to that as non-coding DNA. So when I say coding, I mean coding for proteins. So in this picture, I showed you the coding regions are shown in colors, and we refer to those as exons. And then in this image, as an example of a non-coding region, we have these things called introns. So coding refers to protein producing sections of DNA. Non-coding refers to anything that is not coding for a protein. So what do I mean? Okay, the next slide, you don't actually have to remember any of these, but it's good for you to have an understanding. So you can pause the video and have a reading. These are all different ways that the DNA can be having a function that is not creating a protein. It does maybe sound a little bit complicated, but it does go to show you that there are ways that our DNA is actually being useful to us. So it's not like we've got this evolutionary junk that is not doing anything. So have a read through those if you do want to have a chance. But what I'm wanting to focus on is this thing over here. So what you are looking at is a very colorful, don't normally see them this colorful, a very colorful representation of a DNA fingerprint or a DNA profile. So what you're looking at essentially here is 12 different individuals who have DNA fingerprints that are being created. So what is a DNA fingerprint? So a DNA fingerprint or profile is a visual representation of an individual's genetic composition, so it's a picture to see what they are, which can be used for comparison against a reference sample. So yes, DNA fingerprinting, DNA profiling is something that we can use in forensic investigations, in criminal investigations. But at the same time, it can also be used for things like paternity testing, to find out who the father is, and other applications that have environmental applications. It can take anything that is organic, has DNA. If I want to check the origin of it, that is really what it is useful for. But it's just a much easier than, instead of going and coding your entire sequence of DNA and seeing if it matches, I just use this visual representation. So the important thing is this picture here, the DNA that is on these particular bands, if I could call it that, is actually non-coding DNA. That's why it's important. Because if I was to compare the coding DNA of one human to another human, it would be exactly the same. Because we are both human. If I compared, obviously, between me and a lion, then yes, we completely have different proteins that are being made because I do not walk on 4 legs, have paws, lots of fur, and a mane. That's those proteins that are being made by the lion. But me as a human, if I compared human to human, our proteins would be exactly the same. So then this fingerprint would be useless to us because it would look exactly the same. However, in our non-coding DNA is highly variable. There is lots of variation that occurs in our non-coding DNA between individuals. That's why we share almost the same amount of DNA, 99.9, but we're looking at the difference in that 0.01%. So, how do we create a DNA fingerprint? So this is the process that you're going to need to know. So we have to start with a sample. So either the reference sample from if it's a crime scene or the person, or if it's from a suspect or an individual, we need to get a sample of your DNA some kind. And how do we get that? Well, we can take it through skin cells, hair tags. So there's a little, if the hair gets pulled out, there's a little skin tag that stays next to it. Semen samples, because there's still DNA in a semen sample. Blood, any of those kind of things can be used to create this DNA profile. So the first step after taking the sample is we then need to extract the DNA itself. So this here I've taken it as like the sort of the chromatin, and this is a whole set of DNA. So all the DNA that comes of an individual. But we are only concerned about specific sections of highly variable non-coding DNA. We want highly variable non-coding DNA. So we have to find those sections in this DNA. We have to extract those particular sections. So what we do is we use restriction enzymes. So this is RE, restriction enzymes. So basically they are the scissors of enzymes. So they can go and find and locate in specific areas, and they can sever the link so that I'm only left with these highly variable non-coding DNA sections. Okay, so that's the first part from there. But now that I'm sitting with these small, tiny, microscopic, little, insignificant ones, how do I get to it being like on a picture, on a piece of paper, with those bands that are kind of like that I can see with the naked eye? I'm not looking at it with a microscope. Well, I have to duplicate this DNA. I have to make more of it so that it becomes visible to the naked eye. And what I use is the polymerase chain reaction. You have seen this word before. You have seen polymerase before. It is the thing that makes more of, more of something. So in this instance, we would be using DNA polymerase. So DNA polymerase to make more DNA. So the polymerase chain reaction literally duplicates these sets of DNA exactly. So I'm sitting with much bigger, they put it in a huge vat, lots of the polymerase enzyme, all that kind of stuff, free-floating nucleotides, and they make a whole bunch of these together. So you do need to know about the polymerase chain reaction. Then there are several complicated steps which you don't need to necessarily worry about, but it's basically put through a process called gel electrophoresis. So gel electrophoresis is basically like where you have a machine like this, you have some gel, because it's gel, you put your DNA samples in the little wells, like so one person sample in one well, you send an electrical current through it, and then the DNA bands are separated along this gel slab based on their weight and their electronegativity and all that kind of stuff. Again, it's a slightly more complicated process. You don't have to worry too much about that. On this page, you really only, from a SAGS perspective, you only have to know about polymerase chain reaction. But we use this to separate. And then what we do is we place, once this has been separated, we place a film over it, and essentially we get this picture that looks like this. So this is normally what a DNA fingerprint will look like. And if you look at it, I mean, it's really like, hey, this is quite blurry, hey, and you know, does this one kind of look exactly like this one? Is this the same? It becomes quite difficult. So yes, it is a very complicated, and it needs to be a very precise procedure. But thankfully for you guys, whenever we give you guys questions, we make them a lot easier for you. We try and make it that it's, you know, very definitive in what we're going to give. So please make sure you go back and go through that entire process, make some notes, draw a flow diagram, make sure you know flow diagram of this kind of thing to explain what is required. But just to finish off the video, there are two applied question styles that you can be asked based on DNA fingerprinting. Yes, you can be asked in AIM 1, what is this, describe kind of thing, but from an applied perspective, you can get two types. So the first one is the style of who committed the crime. So it's the example of, I've got someone who's maybe had something stolen from their shop, I have a rape victim or something like that. So we have the victim, we can have DNA from the victim, we can have a sample unknown DNA, and then you will have several suspects normally. And you need to be able to tell who potentially committed the crime. So here's an example. So as you can see, the bands are much easier for us to be able to see. It's much easier for us to tell. So in this picture, I've got the victim and the victim's DNA. I've got two suspects and then two unknown samples. One from crime scene 1, obviously where victim 1 was involved, and crime scene 2, where the victim was involved as well. Now, in this type of question, if it's the who committed the crime, you are wanting to get a 100% match of the suspect to the unknown. Every single band on a suspect must match with every single band of an unknown sample. So, have a quick look. In this, who is the one who potentially committed the crime? So the person who most likely committed the crime is suspect 2. Because let's look. So I've got the victim. I've got two crime scenes. Do we notice that crime scene 2, the DNA from crime scene 2 matches, the bands match only the victim. So the victim must have left behind this sample, whatever it was. Obviously, there would be context for you. So clearly, unless the victim is doing the crime themselves, but we wouldn't give you something as convoluted as that. So clearly, crime scene 2 is of no use to us. And also, suspect 1 has nothing in common with crime scene 2. Suspect 2 also doesn't have anything in common with crime scene 2. So, doesn't really work for us. Crime scene 1, look at suspect 2. These two bands match, even though this and this are side there. This band matches, and this band matches. Suspect 1 sort of maybe matches here, doesn't match this one, and it doesn't match this one. Therefore, all of suspect 2's DNA matches to crime scene 1. So remember, in a who committed the crime, it is 100% matches that you are wanting. So now the other version that we have is who is the father? So when we're doing paternity testing. So now the difference with a who is the father question, in a who committed the crime, you want a 100% match. In a who is the father, you want a 50% match. Now why is that the case? Remember, you inherit half of your genetics from your mother and half of your genetics from your father. So you can't be all the DNA of your father because then you would be genetically identical to your father, which is not the case here. So let's look at this example. So I've got the mother over here, so this is her DNA fingerprint. I have the offspring, this is the offspring's fingerprint, and I have a potential father 1, father 2, father 3. And we need to decide who is the father. So the best way to attack this kind of question is just look at the offspring's DNA. And per band, who is it accounted for? So, pause the video for a sec and decide for yourself who is the father. So, let's go through. Remember, I want to account for every single DNA band of the offspring. So let's look at the first one. Does it match the mother? No, doesn't match the mother. Does it match male 1? No. Does it match male 2? Yes. Male 3? No. Okay, so at the moment, looks like male 2. Band number 2, does it match the mother? Yes. Does it match male 1? Yes. Male 2? No. Male 3? Yes. Okay, so maybe a bit confusing. Next band, does it match the mother? No. Does it match 1, 2, or 3? Matches number 2. Next band, matches number 2. You get where I'm going. Next band, matches the mother. Matches number 1 and number 3. Next one, I've got offspring, between ones from there. So it's still accounted for by the mother, but the ones that are not accounted for by the mother are then accounted for by male 2. So again, this band does not have to match the offspring. It's only that every single one of these is accounted for by either the mother and then the father. So if it's accounted for by the mother, then it's fine, because it can't be the mother and the father. It's either the mother or the father. Last thing is just some AIM 3 considerations that we have. So remember, this is not specific things that you need to learn, but things that you may get applied questions on. You need to have conversations with yourself over this. So what can we use DNA profiling for? Well, we've kind of discussed forensic cases, who committed the crime, getting paternity testing. We could do identifying of human remains if we don't know who they are, illegal trade of things, again, anything that's environmentally based. That's one thing. Then we can look at how reliable or accurate is the process of DNA fingerprinting or DNA profiling. So sometimes this will be given to you in a comparison to other techniques of doing this. You do not have to learn those specifically, but say, for example, I could sequence your entire genome from first letter to final letter and then do a comparison. That would probably be more accurate as opposed to this blurry DNA fingerprinting thing. Plus, there's potentials of human error, all those kind of things. So that, again, it would be a much more situational question that would be asked of you. And the last one, which is really a nice one to start thinking about, is the ethics surrounding DNA sampling. So when I say ethics, is it that I can walk up to you on the street and just say, I need a DNA sample from you? No, I can't really do that. So there's, in a way, you need to be able to argue both sides of the ethics. You need to be able to tell me in what way would it be ethical for us to demand or not, or to be able to get DNA samples from someone without containing consent. So again, if you haven't done anything wrong, you've got nothing to hide. It could be for the benefit of the entire population to create this DNA database. You can catch criminals more quickly. There is a way that you could justify that for the totality of the society. Why would it not be ethical in order to gain without consent? Well, again, if it's still part of your body, it is still your property. I can't take your property without your consent. At the same time, it is still personal information that people are using. Are they keeping it safe? Can someone abuse this information? I don't know. That could be a reason why privacy and consent are important ethical considerations for this kind of thing. Okay, so again, make sure you go through, have a read, make you pause. Again, practice flow diagrams for when you want. Again, if you need some help, I've put my Skype handle in there. Hit me up on Teams. Let's have some meeting. Let's talk about it. And then we can take it from there. Hope you guys are enjoying. Hope the studying is going well.